until today. All right. All right, so let's go ahead and continue on. Everybody doing okay? Nobody too overwhelmed quite yet? Like I said, I've actually have these lectures will be up for you as well. Okay, cool. So what do we got going on? Let's now move into the major muscle groups and the major nervous control of breathing. So what's the major nerve of breathing?
Phrenic nerve.
The phrenic nerve. Is there just one phrenic nerve? What do you guys think?
I thought there was.
Just one? Or do you have two major branches that come off of each side and innervate different sides of the diaphragm?
Yeah.
It's got to be two.
There you go. All right. That's what I like to hear. Okay, so the phrenic nerve, where does it generally come out? What area does it come out?
C3, C5.
Yeah, C3, 4, and 5 keeps the diaphragm alive, and the old saying. So the phrenic nerve is not a cranial nerve. Okay, so certainly high spinal injuries can compromise the phrenic nerve, which means that can compromise our ability to breathe. Okay, so the phrenic nerve primarily innervates what major muscle of breathing? The diaphragm, right? And what happens is the major mechanics of breathing are basically revolving around Boyle's law, right? Boyle's law says that pressure and volume have an inverse relationship. Pressure goes up, volume goes down. Pressure goes down, volume goes up. Fair enough. So, what happens is, when I go to inhale, my diaphragm stimulated by the phrenic nerve, and of course the phrenic nerve is coming ultimately from the respiratory centers, okay, in the brainstem, and the diaphragm drops, right? The diaphragm drops down, contracts, and what happens to the pressure inside of the chest? It becomes lower than atmospheric pressure, right? And Boyle's law says that if pressure goes down, volume goes up. So the diaphragm drops, the pressure decreases, and then there's a gradient, right? There's a gradient. I have higher pressure out here, lower pressure in here, and where's the air going to go? It's going to rush into the lungs, okay? And then when I exhale, the diaphragm relaxes, right? It relaxes and it comes back up. The pressure in my chest becomes positive compared to the pressure outside, and what happens to the volume of the lungs? It decreases, and then air is expelled out to the environment. Is everybody okay there with the basic mechanism? Okay, do you have other muscles of breathing? Did you need something, Paul?
No.
No?
I was just dropping off notebooks.
Okay.
And so forth.
Okay, yeah, if you need to address them or anything, just let me know. I can.
Okay.
All right. You do have what are known as accessory muscles. These are muscles that work in accessory to the diaphragm. What are some of the accessory muscles that we have? The intercostal muscles, right? What else? Okay. You have muscles in your neck, right? Like the sternocleidomastoid or the SCM, the scalene muscles. Okay, so you have these other accessory groups. All right, cool. Cool. So, we've done that. Let me just go back to the transport of oxygen. How is oxygen transported in the body? Okay, what transports it though?
Hemoglobin.
Hemoglobin, right? Hemoglobin. Okay, hemoglobin is a protein. Okay, it's found in the red blood cell. It's a protein that is composed of 4 different units, right? 4 different subunits called heme groups. All right, proteins are all kind of wrapped up on one another, so you have these 4 heme groups, right? 4 heme groups like this. And then within the heme groups, I have what's known as a porphyrin ring. Okay, like this, it's a porphyrin ring. And a porphyrin ring is basically a ring of nitrogen atoms that surround a metal. In this case, what metal is involved? What metal was in hemoglobin? Iron. Yeah, so you have iron. So Fe is a symbol for iron. I'm going to draw iron here. Okay. And then that iron is surrounded in a ring by nitrogens. So I'm going to have a nitrogen here, I have a nitrogen here. Okay. And then imagine, if you will, going into the back of the board. Okay, going back. So you have to use your imagination. I have a nitrogen here, and then imagine popping out at you from the board. I have a nitrogen here popping out at you, okay? And these nitrogens form kind of the backbone of this porphyrin ring. And then the iron down below, imagine down here, is attached to what's known as a histidine residue, all right? So the iron is surrounded basically by 4 chemical bonds, or 5 rather, 1, 2, 3, 4, 5. And you don't have to memorize this, but in what we call coordination chemistry, metals like iron can actually coordinate up to 6 different bonds, which means that there is one site open where something could come in and coordinate with the iron, bond with the iron. What do you suppose that typically is going to be? It's going to be oxygen, right? And if I look at the oxygen molecule, I can find a blue, something with a little more color in it. Let's see if this green works. Okay, so an oxygen molecule actually consists of 2 oxygen atoms, right? And these atoms are double covalently bound to one another. And these oxygens have lone pairs of electrons, all right, like so, all right? And it is actually this lone pair of electrons, because this area is negatively charged, it's more negative than the rest of the atom, because electrons are negatively charged. And we know that the iron is positively charged. And so what happens is the lone pair, one of those lone pair, on the oxygen, I'll draw my oxygen molecule here, is going to be attracted to that iron. So the oxygen will kind of come in at an angle, it'll be attracted, it'll get held in there, and that is how oxygen binds to hemoglobin, right? Everybody's okay with that? You don't have to know all the chemistry behind it and all that. That was maybe a little more detail, but still it's nice to know kind of what's going on a chemical level. You have 4 of these porphyrin rings within the heme groups, so one molecule of hemoglobin can bind 4 molecules of oxygen. Everybody's okay there? All right, we're going to talk a little more about that a little later on when we talk about the oxyhemoglobin dissociation curve, but we're not going to hit on it quite yet. Just building the foundation there. Okay, so that's major oxygen transport, and I'll talk about that in a little more detail here in a bit when we start talking about ABGs, or arterial blood gases. Which did you guys talk about arterial blood gases at least in some rudimentary detail when you took your intermediate or advanced EMT classes?
Yep.
Yes, if you've taken it more recently, it's actually something that we talk more about. If you haven't taken it as recently, you might be more in the dark. At the paramedic level, you will be expected to look at and interpret arterial blood gases. So that's something that we'll talk about today as well, and we'll even do some assignments. ABG interpretation can give people a hard time because it's not quite as intuitive as you'd like to think. But we'll get through it. Okay. Before that, what I want to do is I want to go back to the mechanics of breathing. Okay? There are some capacities and volumes that you guys will be responsible for learning. Okay? There are capacities and volumes. And let me just see if I can sneak in here and grab this. Awesome. Okay. So I want you to imagine yeah, it doesn't have a really, the color is real, yeah, they're kind of, these things have been used and abused. Here we go. Okay, so imagine Yeah, that's a little better. I have a graph here, all right? And imagine that the y-axis is volume. Just imagine this is volume. And imagine that this, the x-axis here, is time. Is everybody okay there? All right? And this is something that can be tough, but you will have to know this. This is basically something you'll kind of have to memorize. So let's talk about this. Imagine that I'm breathing, okay? And I'm going to start right here, okay? And imagine I'm breathing, and I'm just taking normal, everyday breaths. All right? Normal, everyday breaths. Is everybody cool with that? Just a normal inhalation and a normal exhalation. You guys good with that? Normal inhalation, normal exhalation. Then what I'm going to do is, at the end of my normal exhalation, so at the end of a normal exhalation, I'm going to blow out as much air as I possibly can. Are you guys cool with that? So at the end of the normal exhalation, blow, 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 blow. Oh, I can't get any more air out. Okay, inhale. Okay, is everybody cool with that? All right? And then I'm going to go back to my normal breathing. All right? And then what I'm going to do is I am going to inhale as much as I possibly can. And then I'm going to forcefully exhale as much as I possibly can. Exhale, 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 exhale. Oh, I can't do anymore. All right? Everybody cool with that? And then I'm going to go back into my normal breathing pattern. All right. So that is what I just graphed there. Is everybody cool with that? That's what I graphed. Let's fill in what the volumes and the capacities are. What are my normal breaths? Just a normal amount of air that goes in and out of my lungs. What is that volume called? The normal tidal. Tidal volume. All right? Tidal volume. Okay, this is sometimes VT or TV. Okay? Tidal volume. That is just a normal, everyday breath in and out. Is everybody okay with that? Now, what is the maximum amount of air, the maximum amount of air that I can exhale after a normal exhalation?
Exhale.
That's known as the expiratory reserve volume. The expiratory reserve volume, or the ERV. The way that this works is volumes are standalone. When I measure a respiratory volume, it is standalone, but a capacity is when I add one or more volumes, okay? So volumes are standalone and capacities are composite. They are composed of different volumes. Now, is there always a little bit of air left in my lungs no matter how much I exhale? Yes. And that amount of air that's left in the lungs after a forceful exhalation is known as the residual volume, or the RV. Are you guys cool so far? So I have my tidal volume, my expiratory reserve volume, and my residual volume. Everybody's okay with that so far? All right. Now, this is the maximum amount of air that I can forcefully exhale is the ERV. What do you suppose the maximum amount of air that I can inhale is? There you go, the inspiratory reserve volume. Yeah, it doesn't take a whole lot of imagination. You guys got this. So the IRV, the inspiratory reserve volume. Okay? You guys cool with that? Everybody's okay? Those are the major volumes. Okay, the tidal volume, the expiratory reserve volume, the residual volume, and the inspiratory reserve volume. Is everybody cool with that? Now, I am going to have capacities. And what is a capacity? What did I say it was? What is a capacity? How does it differ from a volume? What's the difference between a capacity and a volume?
Capacity is like the most that can be.
No. Remember, I just said it. I just said it.
Like about a minute ago.
I know. Just about a minute ago.
Yeah. I was right.
Capacities are composite. So capacities are composed of multiple volumes. So to get my capacities, I start adding the different volumes together. Is everybody cool with that? Does that make sense? If it doesn't, let me give you an example. We'll go through what the different capacities are. The first capacity that I want to talk about, okay, the first capacity that I want to talk about is the capacity that goes from the, I'll do here, from the end of my tidal volume all the way down to the end of my residual volume. So what two volumes make up this capacity?
The tidal and the expiratory.
Not the tidal, it's the end of the tidal volume. So the tidal volume's not in there. What two volumes?
Expiratory.
In the reserve, residual volume. Okay. The expiratory reserve volume and the residual volume. So ERV, expiratory reserve volume, plus RV, residual volume, equals the functional residual capacity, or the FRC. This is a very important capacity when it comes to intubation, and particularly when we talk about something known as pre-oxygenation. So the functional residual capacity is a combination of the ERV and the RV, the expiratory reserve volume and the residual volume. Are you guys cool with that? Everybody's okay? All right. So that's our first capacity that we'll talk about. The next capacity, okay, the next capacity goes from the top of the IRV here. All right, everybody's cool with that? Goes from the top of the IRV all the way down to the bottom of the ERV. Are you guys okay with that? From the top of the IRV to the bottom of the ERV. Are you guys cool with that? So what volumes are contained within this capacity? Inspiratory reserve volume, expiratory reserve volume, and tidal volume. Okay? So the IRV plus the TV plus the ERV, okay, equals something known as the vital capacity. The vital capacity. The vital capacity, or the VC. Are you guys cool with that? So I have the functional residual capacity, the FRC, which is an addition of the ERV and the RV. I have the vital capacity, which is the IRV plus the TV plus the ERV. Okay. I also have something known as the inspiratory or the inspiratory capacity. Okay. Which goes right here. So this equals the inspiratory capacity, the IC. What volumes go into the inspiratory capacity? Tidal volume plus IRV equals the IC, the inspiratory capacity. And then I have one more capacity, and what is the only thing that we haven't talked about yet in this drawing here? What's the last thing that would seem logical to do?
IRV all the way down to the residual volume.
There you go. Add everything up. Add them all up. All right, so from the top of the IRV all the way down all the way down to the bottom of the residual volume. So this is the IRV plus the TV plus the ERV plus the RV. So it is an addition of all of my volumes, and that equals the total lung capacity, or the TLC. Bam. All right. You guys cool with that? New, kind of probably some new concepts we talked about. All right. You're going to want to know some normal values, and for now, there are really three normal values that I want you to know. I want you to know what the normal residual volume is. The normal residual volume is actually a lot larger than you think it is. It's about, excuse me, it's about 1 to 1.5 liters. So 1,000 to 1,500 milliliters. So 1 to 1.5 liters. And in this class, you will be converting between milliliters and liters quite frequently. So I'll give you the values in liters and milliliters today. I want you to know the normal tidal volume. And you guys should know this. What is the normal tidal volume? There you go, about 500 milliliters. Approximately 500 or 0.5 to 0.6 liters, which is 500 to 600 milliliters. Okay? So your normal residual volume, your normal tidal volume, and then I want you to just know what, in general, what the normal total lung capacity is, the TLC, which is approximately 6 liters. All right, and your average adult, give or take, more or less. Are you guys cool with that? Okay, so these are the volumes and capacities. Everybody's good there? All right. So let's move on, and what I want to do is I want to review. Actually, before I do that, I'll pass these little guys around. So these are the little horror movie that I asked you to. So imagine that I got cut and half of my head went fell off. Okay, so imagine that this is my head that fell off. Of course, that happened. You guys wouldn't be in class, and you'd probably be a lot happier than you are now. But just imagine that happened, and then you can kind of smile to yourself. All right, so in here I have a little brain, and this just kind of allows you to visualize a little better. So the this part right here is the brain stem that I'm looking at. This is a brain stem. This is a cerebellum. This is the upper brain, or what we call the cerebrum. This is the corpus callosum, which connects the two hemispheres. Okay, so the brain stem right here, and this little area that bulges out of the brain stem, that's the pons. Right in here, that's the pons. And then this little bulge right underneath, or just inferior to the pons, is the medulla. So I figured I'd just pass these little brains around for you guys to take a look at. And I figured I'd pass these little guys around as well. Okay, so imagine that the top of my head got lopped off and it fell down. And you're just looking at my airway, okay? Right? Okay. So this is anterior, and this is posterior, and this is a view of the glottic. This is basically that terrible picture of the glottis that I drew a little earlier, where I have the tongue. I have the tongue here. I have this little valley in here, the vallecula. This little flap here is the epiglottis. And then you can see that deep in there I have the vocal cords, the vestibular folds, and you can actually see the cartilage quite well. Remember, the big bulbs are the corniculate cartilage. The smaller bulbs toward the midline are the arytenoid cartilage, the arytenoid notch, the glottis, and then you have the posterior. What's this posterior structure here, this hole? It's your stomach hole, right? You got your stomach hole here, and you got your lung hole here. So I'll pass these little guys around. You can look. I also have the major cartilages of the larynx. What's this big thick cartilage here? This is the thyroid cartilage. What's this little valley right here? Cricoid membrane, and then what's this cartilage here? Cricoid cartilage, and the whole thing is called the larynx. or the voice box. So I'll hand that to you guys and you can take a look. Okay, everybody cool with that? So that's just some visual aids to break up the monotony of the lecture a little bit. Okay, let's talk about breathing patterns real quick. Okay, breathing patterns. So you have different breathing patterns, and you will have to know these, and you'll have to identify these on tests. And they're not going to give you a picture. They're actually just going to describe it in words. So you need to be able to describe the pattern in words as well. But let's just say that I have a pattern of breathing that looks like this, with a normal tidal volume that occurs about 12 to 20 times per minute. What would we call that breathing pattern? Normal, right? Just a normal breathing pattern. Okay? What is another term for normal? It's a weird kind of term. That means everything's good to go.
Okay.
It does mean A-okay. It's U. Remember that EU. U means good. EU. So eunypnea is a normal breathing pattern. Eunypnea. Okay. Now, if I were to see this occurring greater than 20 times a minute, we call that what?
Hyperventilation?
No, not hyperventilation. It may cause hyperventilation. Tachypnea, right? Tachypnea. Tachypnea, right? Just basic medical terminology. And then if it was less than about 10, breadnypnea. All right, breadnypnea. Yeah. There you go. You guys good with that? Now, tachypnea may cause hyperventilation, but tachypnea isn't necessarily hyperventilation. Hyperventilation is a condition by which I blow off too much CO2. Okay, that's what hyperventilation is. But the actual breathing pattern, I'm not talking about what's being ventilated, how much air is being going in and out of the lungs. I'm actually talking about the breathing pattern here. So tachypnea, breadnypnea. Everybody's cool with that? And then eunypnea or normal. I have several abnormal patterns that you want to be familiar with. The first is associated with acidosis and acidotic states. And it's very rapid and very deep. And what is the name of that pattern? Kussmaul's respirations, right? Kussmaul's respirations is strongly associated with acidosis. Okay, DKA. A lot of people say DKA, but metabolic acidosis in general, not just DKA. We have another pattern that looks very much like Kussmaul's. In fact, I'm not even going to draw it because it's very similar. But it is not due to acidosis. Rather, it is due to some sort of brain problem. Maybe there's brain trauma or swelling, increase in cranial pressure. And it looks just like Kussmaul's. It's very similar to Kussmaul's. They breathe very deep and very fast. But that pattern is known as central neurogenic hyperventilation. Okay? The difference is acidosis causes Kussmaul's, and some sort of neurological, some CNS problem causes central neurogenic hyperventilation. There's a really good video on YouTube. You can go and search for it, where it's a young guy who's skateboarding, and he tries to go down some stairs, and he slips, and he smashes his head down, and his friends run to him, and he's actually unresponsive, laying on the ground, and he's going unresponsive after smashing his head, and it's just a perfect demonstration of what central neurogenic hyperventilation looks like. Very similar to Kussmaul's. Okay, there are some other ones that are a bit harder to talk about. Okay, the first one,
it was central neurogenic what?
Hyperventilation.
Hyperventilation.
Yeah. So the first one is where I'll have these periods of apnea, and then what will happen is I'll have these little periods where my tidal volumes start out small, and then they get bigger, and then they get bigger, and then they become smaller and smaller, and then I have a period of apnea, and then I do it again. Say it. Cheyne-Stokes. That is Cheyne-Stokes respirations, right? Where I go. Just like that. Okay. That is Cheyne-Stokes respirations. There is another pattern that is somewhat similar to Cheyne-Stokes, where you don't have as much, you don't have this pattern of small to high, back down to small, where it's, you still have these periods of apnea, but you kind of have these periods of, you take a few breaths, and then apnea again, you take a few breaths, but the variation, that cyclic variation is not present like Cheyne-Stokes. And what is that known as? So this is Cheyne-Stokes here, and then this would be, starts with a B, Biot's respirations, okay? You also have what's known as agonal respiration, where it's just kind of an occasional gasp a couple times a minute, okay? And then you also have what's known as apneustic respiration, where you have these pauses. You have these pauses followed by a large, long breath. And then another pause and a large, long breath, another pause. Okay, that's known as apneustic. So we have Cheyne-Stokes, Biot's apneustic, Kussmaul's central neurogenic hyperventilation, eupnea, tachypnea, bradypnea. Okay, these are some of your major respiratory patterns. Kussmaul's respirations are generally caused by acidosis, right? Metabolic acidosis. These guys here are generally caused by some sort of neurological insult, a stroke, could be some sort of neurotoxic drug, okay? But those are generally the Biot's, the Cheyne-Stokes, the apneustic are generally some sort of nervous system problem. And then your agonal, just your occasional gasping, that's generally dead. Okay, generally dead people do that. You know, when you're, have you ever coded somebody where they don't have a pulse, but they still have an occasional gasp here and there? Yeah. Okay. You guys good? You good with that? All right. You want me to leave this up here, or did everyone get this? Okay, I was going to erase it because now it's time to talk about acid-base balance, oxygens and carbon dioxide transport. And this is where it becomes perhaps a bit more confusing for some people. Now, if you're a recent graduate of the advanced EMT curriculum, you've actually had some acid-base theory. If you are a graduate of the older state EMT intermediate program, you likely have not had a particularly robust experience when it comes to acid-base fluids and electrolytes. That's why we also supplement your reading with the acid-base fluid electrolyte book. But let's go ahead and see if we can talk about this stuff. All right. So, when we talk about gases and the transport of gases, gases are normally what we call non-polar molecules. Okay, anyone who, most of you guys should have taken my chemistry course that was integrated into the pharmacology. I think a lot of you guys were in that last semester, right? Yeah. So, what's the difference between polar and non-polar? There you go. Where the charge is, where the electrons are. In a non-polar molecule, the charge is spread out more or less evenly. Okay? There are no single area of the molecule is more charged than the other. The charge is spread out more or less equally. Okay? In a polar molecule, you have separation of charge, and you have areas of that molecule where the charge is more concentrated, where you have electron density that's more concentrated. So an example, the best example I give of a polar molecule is something like water, which is H2O. So you have your oxygen that is single covalently bound to two hydrogen atoms. And we know that the oxygen is a hog. It's an electron hog, right? It really likes to get a hold of electrons and hog them and hold on to them. And it doesn't like those hydrogens to have those electrons. It's like, I'm not really going to share too much with you. And that property of oxygen is what we call electronegativity. It's highly electronegative. It likes to hog those electrons. So what happens is you have more negative charge around the oxygen there. And because the electrons are being pulled away from the hydrogens more, you have a more positive charge around the hydrogens. Is everybody okay with that? And that's what makes this molecule polar, is the charge has been separated. You have more electron density here, so this area is relatively more negative, this area is relatively more positive. But that's a good property for water to have, because when you have other charged substances near water, like sodium, what is sodium? What's sodium chloride? NaCl. Okay, sodium and chloride ions. What are they going to do when I put them into water? You guys have seen this, right? You put salt in water, what happens? They get dissolved, right? I put sodium chloride salt crystals into water, and they dissociate, and the sodium is attracted to the negative part of the oxygen, and then the chlorides are attracted to the positive hydrogens. And water is really good at dissolving stuff. Polar substances tend to dissolve very well in other polar substances. Okay, that's a good rule of thumb. Let me ask you now about gas like oxygen and carbon dioxide. So oxygen is two atoms, two oxygen atoms, double covalently bound to one another. Is this a polar molecule? What do you think? No, it can't be, right? Because if it was oxygen and something less electronegative like hydrogens, yes. But here it's just two oxygens. They both have the same electronegativity, right? So they're going to have equal sharing. And the electron density is going to be spread out more or less equally through the molecule. And you can see that this molecule is flat. See how it kind of takes on a flat appearance? Whereas the water has a more bent appearance. And the bendy appearance is because it is characteristic of something that's polar. So oxygen is nonpolar. Let's look at CO2, carbon dioxide. Okay, that's a carbon here in the middle. Okay. And it is double covalently bound to two oxygens. What kind of shape does CO2 have? Is it bent like this, curved, or is it flat? And what's the general rule of thumb when it comes to molecules that are really flat like this? Non-polar. Both oxygen and carbon dioxide are non-polar molecules. Okay, stuff doesn't dissolve in, or we'll reset. The general rule of thumb is non-polar molecules do not dissolve well in polar solvents like water. Okay? That's a disaster for the human body. Would you guys agree? That's an absolute disaster. Because think about this. Just think about this. I need oxygen to get oxygen to my cells, right? What does oxygen have to be transported through to get to my cells? Blood. What is blood primarily composed of? Water. A disaster, a freaking disaster. So what has happened is through evolutionary mechanisms, okay, billions of years, we have evolved, the proteins that have evolved, transport proteins, and that's where we have hemoglobin. Hemoglobin is a transport protein. Lots of different animals, it's not unique to humans, lots of different animals actually have hemoglobin. Some animals actually have proteins that are different, like spiders. Some types of spiders they have molecules that are like hemoglobin, but they're different. They use different metals, like I think like zinc and copper and things like that. But what we have to do is, we have to do one of two things. We have to attach these nonpolar molecules to something in the blood to transport them, or we have to turn those nonpolar molecules into something that is polar. Does that make sense? Those are the two ways. That's what we have to do to transport O2 and CO2. So what I'm going to do is I'm going to draw you guys a little diagram here. I'm going to diagram out the we're going to talk about what's going to go on in the lung. So here I have the I wish these things were a little darker. An alveolus in the lung. It's just not very dark. Here we go. So I have an alveolus in the lung here. All right. And then I have my blood flow. And then over here, all right, over here, I have tissue. So these are my distal tissues. All right, so imagine, if you will, this is some sort of cuboidal tissue. Okay, whatever. It's terrible, but just use your imagination, okay? I've got tissue here. I've got my alveolus here. All right. So let's talk about what's going on in the tissues. What are the tissues producing as a waste product? These guys are producing CO2, right? They are producing carbon dioxide, CO2. Everybody's cool with that? All right. And then what do I have here in my alveoli? Primarily O2, right? I also have a lot of nitrogen, but we don't really care about the nitrogen. It doesn't. Actually, most of what we breathe is nitrogen, but we're more concerned about oxygen. Okay. So I have got to get this O2 all the way to the cells, and I've got to get the CO2 all the way into the alveoli. Is everybody okay with that? That's what has to happen. So there are a series of chemical reactions that happen. Let's talk about oxygen first, because it's fairly straightforward. Oxygen crosses the alveolar capillary membrane, right? And it is attached to hemoglobin, right? Everybody's good with that? Hemoglobin plus O2 equals O2Hb, or oxyhemoglobin. You guys good with that? And then the oxyhemoglobin is transported over here to the tissue, where the oxygen is offloaded, okay? And the oxygen is offloaded into the cell, when the cell needs it, all right? And then you have deoxyhemoglobin, and the deoxyhemoglobin is circulated back to the lungs, where it picks oxygen back up, drops the oxygen off, picks it back up, rinse and repeat ad infinitum. Everybody's okay there? Cool. CO2 is a bit more complicated. The primary way by which CO2 is transported in the blood, a little bit of the CO2 can attach to hemoglobin, to deoxyhemoglobin, a little bit. It's called a carbaminohemoglobin. But most of the CO2, we actually have to transform it into something polar. So this is what happens. CO2 at the level of the tissues, okay? CO2 is combined with H2O, okay? So H2O plus CO2, and there's an enzyme that makes this chemical reaction more efficient, okay? And that enzyme is known as carbonic anhydrase. And it should talk about this in the book, right? In your gas transport, when you get to that reading. So carbonic anhydrase catalyzes or makes this reaction more efficient. And what that does is that gives me a molecule of H2CO3. Okay, because I have two hydrogens here, right? Right? I have one oxygen here and two oxygens, so that's a total of 3 oxygens and 2 hydrogens. 2 oxygens, 2 hydrogens, 3 oxygens, and a carbon. Everybody's cool with that? So I have all my different atoms. This is what's known as carbonic acid. You guys cool with that? Carbonic acid? Carbonic acid is really unstable in water, however, and it breaks down. And it breaks down into hydrogen ions, H pluses, and bicarbonate ions, HCO3 negatives. Now, are these polar?
Yes.
Yeah, they're charged, right? If you're charged, you're polar. So the CO2 is turned into bicarbonate and hydrogen ions, which means that these can then get dissolved in the blood, and these can be transported by the blood to the lungs, okay? Where in the lung, I have carbonic anhydrase again, and what it does is it helps recombine these two, okay, to give us H2CO3 carbonic acid, which then breaks down into CO2 and H2O, and then the carbon dioxide can then be eliminated. All right, are you guys cool with that? That's how most of the carbon dioxide in our body is transported through this mechanism. Okay, so I have CO2 and O2 transport. Are you guys good with that so far? Is everybody okay? You're so quiet all of a sudden. You guys are so quiet. Everybody's more or less okay with how that mechanism works. Okay, cool. I'm going to talk about an effect real quick, and then I'm going to get you guys on a break since I've been blabbing on for a while. And then after the break, we'll start into arterial blood gases. All right. So, when we talk about hemoglobin, don't you guys think that a natural question is, how does the hemoglobin know to release oxygen? Isn't that a natural question? What do you guys think? I would ask that question, like, okay, cool, hemoglobin transports oxygen, but what gets the oxygen off the hemoglobin? What makes it do that? Well, what happens is there's something known as the Bohr effect. The Bohr effect. And it is a rather ingenious mechanism. What happens is, what do you have a lot of around the tissues? What do you have a lot of? A lot of CO2 becoming carbonic acid, a lot of carbonic acid becoming hydrogen ions. Would you guys agree? So what would you say about the pH right here around the cells versus the pH here in the lungs? If you had to guess. The pH is going to be more acidotic. It's going to be lower around the cells than it is in the lungs. So here's kind of what happens. So imagine that I got my O2 hemoglobin, my oxyhemoglobin here in the lung. All right, and then it gets transported down here to the tissue. And I'm just going to draw a bunch of hydrogen ions here. All right. Because I have CO2 being converted into hydrogen ions. So what's going to happen is here where the pH is high, it's relatively high, it's more alkaline, oxygen will bind hemoglobin, and then as that hemoglobin travels through more acidotic tissue, that pH, the low pH, the decrease in pH, is going to change the shape of the hemoglobin ever so slightly. What's called the conformation. It's going to induce a conformational change. And when in the presence of low pH, hemoglobin will release its oxygen to the tissues. That's a really ingenious mechanism there. And that is known as the Bohr effect, B-O-H-R, the Bohr effect. Christian Bohr was a Danish physiologist who actually came up with this. Anybody here taken chemistry, like the actual chemistry? Do you remember something called the Bohr model of the atom? Do you think that was him? Actually not, it was his son, Niels Bohr. So a pretty, I bet there's some really interesting tabletop conversation when the family got together to eat. You have this physiologist who figured out deep, and I'm talking, this is back in the 1800s, of the late 1800s, when Christian Bohr figured this out. Niels Bohr, of course, the early 1900s is when he came up with the Bohr model of the atom. All right, cool. You do want to know this equation here, though. Okay, you're going to want to know this equation. You're going to want to know what carbonic anhydrase does. You're going to want to know that CO2 plus H2O can become carbonic acid, what breaks down. And this chemical reaction can work this direction and this direction. That's what these arrows mean. This is a equilibrium reaction. It can occur both directions. Okay. So it occurs in this direction in the tissues, and then it occurs in the opposite direction when you get to the lungs. Okay, where hydrogen bicarbonate ions are turned back into CO2 in the lungs, and in the tissues it's CO2 to H plus and HCO3 minus. All right, guys, take a 10-minute break.